Thank you for inviting me first. It's a great pleasure to talk here. What I'm going to show you is really preliminary work. We just, let's say, every time we start looking at this system, we end up getting more and more technical problems. We end up developing more software. So there was a lot of the things that Zen already showed. That was kind of the beginning of what I'm going to show you today. So this was supposed to work. It was working before when I tested. 1 second. Okay, it's not working. My first contact with this respiratory system was when I joined Klaus Schulten group, and a lot of people there working on this photosynthetic systems and photosynthetic apparatus, and also on the respiratory system. Klaus actually had work already on the complex 5, the ATP synthase, also in the complex 4, the cytochrome C oxidase, and the BC1 complex, and got a few papers on that. But he never had a chance to work on the complex one, actually. He fortunately passed away when you were starting to do this work. And that's the main part that I'm going to talk to you today. But like, how does this complex work? What happens here is to generate energy in your cells, or also in the bacteria, you need a lot of different systems that do a different step they do different steps in generating ATP. So what they do is they basically pump hydrogen to outside of the cell at the same time that they are generating a potential that can can move ATP synthase. Some of these complexes, like complex 1, 3, and 4, they are known nowadays to form what they call the respirosome. So basically they form a super complex that they can interact with each other more easily. So the most studied system is then, the most studied respiratory chain is then the eukaryote one. That what you see is you have the complex 1, this is the first step of the reaction, and you have NADH being converted to NAD plus a hydrogen, and then you have a chain of reactions occurring that takes a transport 4 protons across the membrane, and a quinone being converted to a quino. This quino can come and work also together with the cytochrome C2 that can also pump another 4 protons. And then another quino converted to quino come to the, just gets up. So then the cytochrome C comes to the cytochrome C oxidase that also sends 2 protons across the membrane and also converts oxygen to water. And then by this potential that you have, you enter protons, and then the ATP synthase generates ATP. So the part that I'm going to talk today then is the complex 1. So the complex 1 is the largest of this, the respiratory complexes, and it was the most, it was just solved recently. Actually, the structure of the other components are known for quite some time already, but the complex 1, the first structures came out in the late 2000s, like only parts of the system, and the complete complex 1 came out in 2013, the first structure. So what this first structure does, so basically it couples the substrate oxidation and the hydrogen reduction to activate the proton transport. And when you see this system is quite large, the smallest version of it is in the bacteria, has 550 kilodaltons, and the human one, for instance, has about 1 megadalton. But the necessary conserved subunits are 14 subunits that are present in the bacteria. If you remove the other subunits from the human, it still works. So basically, actually, people don't really know what these other subunits do. And you need only these 14s to make the system work. The first structure then came out is the one that we are using, came out in Nature in 2013 from the Sazonov group. And it's from Thermus thermophilus. There is also like one structure that came out later from 2014 from bovine, and then from ovine. And the human respiratory complex came out in 2 separate parts, like one part of the, I'm going to talk about the membrane part and this peripheral arm last year, and this year they came out the complete respirosome. But all these structures here, they have quite low resolution except for the first one. So this one is a crystal structure, have like a 3 angstrom resolution. The other ones are all cryo-EM, so the resolution is lower. So for us, the bacterial one is much better to develop our studies. So that's why I'm going to talk more about that. So how this structure is divided, you have a peripheral arm where a chain of reactions occur. I'm going to show you a little bit more. And then you also have, so basically this peripheral arm comprises this NADH oxidizing step and the quinone module. And then you have the membrane arm where the proton transporters work. Also, some of these proton transporters are actually antiporter-like transporters. So if the channel is deactivated, they also work as an antiporter, so they can transport sodium to inside the cell and a proton to outside. So the main part of the respiratory chain is, let's say the most complicated part, I would say, is the process where you have NADH coming, and then you take this electron from the NADH, then you have this electron transfer reaction occurring that goes all the way to the quinone. This is quite far, and actually, oh, it's the next one. So it goes through a chain of iron-sulfur clusters where this electron goes through, and this, oh, I forgot to, it's actually later. Okay, forget it. So the distance, I can just tell you, I'm sorry. The distance between the NADH and the quinone is about 96 angstroms. So it's quite far for electron to be transported. Some of the jumps here between these iron-sulfur complexes are 14 angstroms, which is kind of in the limit that they say you could do it. So this peripheral arm then has this NADH side. This is actually a flavine and an NADH. And then you have also the quinone and the iron-sulfur complex chains. So the first step of this reaction, then NADH comes and also flavine, and then you reduce flavine, and then you oxidize NADH, and then you leave one electron, extra electron. This electron then jumps across these chains of iron-sulfur complex. Then you have a quinone that's converted to a quinol, and it's what's supposed to trigger the next step I already mentioned, like it's 95 angstroms apart, and it actually triggers these proton transporters across the membrane, and then quinol leaves the site. So what is believed to be the longest step here is actually to quinol to bind and unbind. So that's probably what takes longer in this process. Okay. So what are the biological questions we have? There are many questions that are regarding the complex 1 itself. For instance, how exactly is the coupling between the electron transfer process in the peripheral arm and the proton channels in the membrane arm? Also, there are many questions that are like how they work together as a respirosome, like we're seeing now. So, but that's a lot of diffusion questions, how quinols diffuse from one another and how they interact, and what is the time of this process to occur. So to study some of these systems, we actually start looking more in detail of this structure, and we start to see also some different things about the structures. For instance, inside the transmembrane arm, you have this antiporter-like channel that is connected through a 110 angstrom helix across the membrane that connects another part. So it seems like they are connected, and we don't know. Actually, we start reading about it. It's actually one of the main questions is you have 4 proton transporters, pumps actually, and they work in parallel, but like, are they going at the same time? Are they going like in a chain? How exactly they work? It's like one opening after the other. And so we decided to look at these 4 proton pumps. But then we came to the problem that Zen already mentioned, that if we want to look to these proton pumps, we're going to have to look using quantum mechanics, because we need QMM methods to simulate how the proton pumps, how the proton is jumping and breaking bonds in the simulation. This is quite a large QM region if you want to do QMM simulation. That's basically impossible, so we could not do it. So then our solution came by thinking, okay, we might do, instead of doing 1 QM region, we can make like 4 QM regions. Each one of these transporters actually, it's an independent transporter, but that is seeing the other one as an MM region. So that was the solution we found. And actually, by doing that, we could just go ahead and make 18 QM regions, like we could just make, 16. We could make every single part of this protein that it's either having a reaction or it's harder to simulate, let's say, because you have coordinations and things like that, the classical force fields are not so good. And then you can just make all of them QM because the cost would be the same. You can parallelize that. So then we went ahead and worked on developing this QM interface in NAMD. Well, Zen already showed it a little bit, so I'm going to kind of jump. But basically what Zen showed you is this version where you have one QM region, and this QM region is sent, like the atoms are sent to ARCA, for instance, and then the MM are handled by NAMD, and there's some communication between them to calculate how one part of the system fuels the other part of the system. But now, let's make something more complicated. What if you have two QM regions? So you have to be sure that one QM region is seeing the other one as an MM region. So basically what I'm telling you is, when we do that, ARCA, or any QM code you're using, takes one of these regions and look at it as, let's say, the QM atoms, and all the other atoms are point charges in space. So basically just feeling the potential from the charges of those atoms. The same thing is valid for the other QM region. So one QM region is seeing the other as just charges in space, like it's seeing the MM atoms. So you calculate the gradient over those atoms, send that back to NAMD, and NAMD does the integration step, moving the system ahead. So that is done. So then you can see, you can basically make all these regions to work in parallel, and it's basically like seeing another MM part of the system, but you're seeing another MM part of the system where the gradient of the forces are calculated through a QM potential. So the same idea can be used for many things. So like the development of this code was not limited to what we are doing. So you can use it to study enzymatic reactions. You can use it to study the setting of the genetic code, like Zen showed. You can also use it to study whole proteins in the QM region. So this is an entire protein in the QM level. Or also you can use to study protein mechanics. That is something that I'm interested on. So all of that, back to our main problem now. So we can now try to use all these methods that we developed to the main system. So again, we are back to our system, the 3.3 angstrom resolution we have for this thermosthermophilus complex 1. So then we start to do all the normal protocol for MD simulation. So we solvated the system, 2.5 million atoms. We added about 3,000 lipids, and sodium chloride is the size of the system. We had like about 360 angstroms squared plus 220 in Z network, it's shorter. And then we did 100 nanoseconds of MD simulation equilibration. And this part we are still running some of it, like about 1 microsecond of MD simulation without constraints. And then we started the QMM simulations. So I'm going to show here now is the first part of the step. So for these reactions to occur, the first thing you need to do is, we kind of know the residues that are important for these reactions, for these proton pumps to work, but you need to hydrate them close there. Otherwise, they would not get the first proton, the first step of the reaction. So they need a hydronium in that area that gives the first proton and then starts the whole process. So when we do a simulation, we can start seeing like this being hydrated. This is water molecule, and you can start see the forming of some pores here. So these pores are the pores that we have to transport part of the to transport this hydrogen. What you have in the middle is like a lock that water cannot go through, but the proton can jump through. These are histidines and lysine residues where the proton can jump through and make the proton pumping process. So from this point, we then create our QM regions. Each one of these regions has about 500 atoms. And then what we got is this. This is just new result. We just got it. So this is the first time we are doing this system with 4 QM regions. And this is like an initial simulation where you're not forcing any reaction. We're actually forcing it to stay in place just to minimize the energy. And you can see already atoms moving here. And this is like already in QM, and with 4 different QM regions working side by side. So what's next? Our next step here is to start forcing the reactions that we believe are happening. So the same way that we did for the synthetase, like Zen showed. And by doing that, so here again you can see better all the atoms moving. Some are blinking here because of the way I rendered the video, sorry. But what happens is like we are gonna force the pathways that we believe are possible. And by doing that, what you have is an initial part where you can start your string calculation that I'm going to explain a little bit. But basically, if you have the same initial and final condition, you can optimize the path. And that's what we're trying to do now, next. And then calculate free energy. So what we wanted, of course, was something like as simple as this. Like I'm showing here, these are just hydroniums in water. You can easily see like they're forming bonds and unforming, but that's not as simple as what is going to happen in this system. So the first step is to do with steer molecular dynamics, where we force reactions, like in sequence, the way we believe they can happen. And then we can use a string method to optimize this minimal path. So what we do here is we basically force the system to find the minimal barrier possible, like kind of like a valley in the mountain region, you know, like you usually see these images, to find this valley so where the reaction is easier to occur. And then we can calculate free energy over that path. That's our main goal. So the first step, then again, forcing these reactions to occur, we created a special SMD for this kind of thing in our QMMM code, where we can stop the SMD step as soon as the reaction occurs, and the system can just relax after that automatically. We then, after doing that, we can then get the strings. One of the questions we have right now is, will this regular way of doing string calculations with swarms of trajectories work for this kind of system? Because here you have 4 protons, 4 channels working side by side. Will the string method distinguish which one goes first, or if they go all in parallel at the same time? We don't know for sure if that's going to work or how much computer time you're going to need to get these answers. But that's the next step. And then as soon as we get a string, the next step I would say is relatively easy. That is to get the free energy over those paths. And that's our main goal for the future. So what we want at the end is to understand how exactly this electron transfer and proton transfer are coupled. So like how exactly these two parts of the system work together, and also get, hopefully, the rates of these reactions and see how we can modify them, play with these numbers. And with that, I would like to thank everybody that helped. This is kind of the same slide that's essential. All these people that help us to develop the QMMM interface. And you all, if you have any questions.
Thank you very much. Any questions?
So you've broken down your system into multiple QM pieces.
Yes.
And you're reliant then on the MM to couple those.
Yes.
So do you try to keep your QM regions about the same in each case, or do you try to? So how isolated are these QM pieces? I guess that's
Okay.
the question.
So to select those regions, we actually, I can let in the video here. At the end of the video, I have it. Kind of easier to see. So to select these regions, like I know already some of the residues that are important. I know for the literature that are important for the proton translocation. So what I did was to select all the nearby residues so that you have sort of like a channel where they can go through, and also the nearby residues to make the, let's say, the polarization around those residues correct. The only thing that you have to do is actually the way we develop the code. It's impossible. It's like to do it wrong, is to make sure that every one of these atoms is just in one QM region. Otherwise, you would have a problem. But the way we break the system, it's impossible to not do that, because they're really, really next to each other in this case. So
Yes, this is actually a related question. So you need to include the water as QM region, right?
So
then.
yes, in this case, I have some water molecules.
And they come in and out. So does that affect?
Yeah, so I'm not showing them here because I had to reduce the size of the file to render the video. But I can do that later. That's, so yes, water can go in and out. This simulation that I'm showing is quite short. It's 5 picosecond, which is a lot for QMMM, but it's quite short, so we don't see water leaving. We created a way of replacing water molecules in MD. So if one water goes out, you can have another molecule replacing that one. It gives you, it's not the best way of doing it because of the potential energy, you have some kind of jump when you do that. But if you're looking the free energy space, it's okay, like we are doing. So yeah, you need the water molecule, you need everything. So you need to make the whole channel. So at the end, what I estimate is that each one of these QM regions is going to be like about 1,000 atoms, like my final version where I have the SMD going. So that goes kind of for the next point, that is, you can only do that in semi-empirical right now to do all these calculations.
So these are semi-empirical calculations?
Yeah, these are all semi-empirical. Right now, the way we are doing, I would say it's impossible to go beyond that. Like maybe with TerraCam we could go, but it's pretty heavy what we are doing.
Any other question?
I'm going to introduce you.
Thank you very much. Otherwise,